Go back through, we did the first 6 on Tuesday. Let's going to go back through them again. 132. And we're supposed to do both cranial nerve activities and the brain dissection today, but we don't have time to do the cranial nerve activity, so we'll do that tomorrow. Most of it, in fact, you can do at home. In fact, all of it you can. Swallow coffee and where do you taste it and follow a pencil around. It just helps to have somebody do that with you, okay? So I'll have some of the material here tomorrow that you can work on. The morning section didn't quite finish. Some of them found the hippocampus, some of them didn't have time, and so we saved their brains aside, and they're going to work on that tomorrow. So it depends on how far you get today in the brain dissection. If you want to save your halves of brains and do some more dissection or following coronal sections or something on your own, you may do the same as well, okay? But the cranial nerve activity, we're not doing today. We're just doing the brain dissection in lab today. And then the cranial nerve activity will start tomorrow, and you can either finish tomorrow or finish at home because it's, you know, swallow cold water, move a pencil around and follow it. So that kind of stuff. I'll go over the activity with you, but you don't have to do all of it in class tomorrow. All right, so since Tuesday, you've all memorized the on Olympus towering top, or Gretchen's favorite one, which I'm not going to say again. And so I want to go through these cranial nerves, the first 6 again, and go into a little bit more explanation of them. And we'll take a break kind of about cranial nerve 9 or 10, and then come back and finish the cranial nerves and then kind of tie them together as far as how I might ask questions or how you can group them together, okay? So that you're not feeling like they're all just 12 disparate different neurons. So remember we had these 5 different modalities, and we have afferent on this side. So these are the 3 different types of neurons. So we're talking about nerve cells carrying these 3 different kinds of information. And there are 3 actual different neurons, all right? Our neuron doesn't carry pain, touch, and pain one time and taste another. So they're distinctive and always carry that same kind of information because remember it's being processed by where that pathway ends up in the brain. So if you stimulate a pain receptor and it ends up in a touch receptor, you're not going to feel pain. I mean, a touch processing, you're going to feel touch. All right, so the pain neuron is always going to carry pain. The touch neuron is always going to carry touch. And so these 3 different modalities for sensory are general somatic, pain, touch, and temperature. Different neurons carry each kind, but they're classified as general somatic afferent. Pain, stretching from organs, pressure maybe from organs, an ulcer pain, heart attack pain, too much gas is going to be general visceral afferent. And special visceral afferent, remember, is our special senses: taste, balance, vision, hearing, smell. Now, what is the type, what is the shape of the neuron that we find in the spinal nerve or cranial nerve, the peripheral nerves, that carry sensory information? Remember we talked about 3 major shapes or appearances? Bipolar is sensory, but it's not what most of them are. Pseudo-unipolar, that's what forms our dorsal root ganglion, are the cell bodies of the pseudo-unipolar, all right? So these will be pseudo-unipolar neurons, which are shaped like this. And the ganglion is found outside the central nervous system. All right, so this would be our sensory ganglion. In the spinal cord, we call it the dorsal root ganglion. In the brain, they each have their own name. All right, so the trigeminal ganglion is known as the semilunar, facial is known as geniculate, and so on. But it's a ganglion because it's a collection of cell bodies outside the central nervous system. All right. Special visceral is also pseudo-unipolar except for 2 special senses: smell and vision, and those are bipolar. So our olfactory and our optic nerves have bipolar neurons instead of pseudo-unipolar neurons. And I'm going to diagram that pathway for you to illustrate. Okay? But the other special senses would have the pseudo-unipolar that we see with pain, touch, and temperature as well. So we looked at this briefly on Tuesday, and we haven't really gotten to senses, special senses, or any senses yet until tomorrow. And so in our sensory pathway, we always have a neuron that is a receptor. And I've just kind of done a diagrammatic here. So we have in the skin, we talked about light touch receptors, which were the Meissner's corpuscles, and pressure receptors, which were the Pacinian receptors. All right, when we get to physiology, this is for free right now, just to kind of lay the background. When we get to physiology, when a receptor binds a smell particle, an odor particle, all right, or we touch it lightly if we're doing the Meissner's corpuscle, it creates what is known, and this is for free until our next unit, it creates what is known as a graded potential. Graded means it could be weak or it could be strong. So to illustrate, if I throw a small pebble into a large lake, it's going to, bless you, it's going to create waves or ripples, all right? If I throw a bigger pebble or a rock, the biggest rock I can pick up, it's going to do the same thing, but the waves are going to be larger, okay? So that's a graded wave, or graded amount of energy. Now, if I'm at Lake Tahoe and I throw a boat into the lake, it's going to create a huge splash and big waves. Will those waves reach the opposite end of Lake Tahoe? Probably not, because that energy will dissipate. It'll be absorbed by the rest of the water, okay? So the energy, the current, the electrical current that enters into the receptor may not be strong enough to actually cause the release of chemicals to activate our actual sensory neuron. And that signal may never reach. So I might be actually touching little hairs on my face, but I'm not activating the receptors because I'm not creating a strong enough current, therefore that signal never actually gets to my brain. Okay?
Are some people more sensitive to, so that the difference?
Right. And adaptation occurs too. So when I have this lying on my hand, or if I'm wearing, a sweater or whatever, I may not actually send those signals to my brain all the time, or if I do, the brain doesn't process them. So we have adaptation that occurs as well. So some people adapt sooner, some later, some are more sensitive to the levels of chemicals that are released. All right? So let's say this is my receptor for smell. And chemicals that I'm in the air that I'm smelling will bind to that and may or may not release enough transmitters to activate my bipolar neuron. But if they do, and enough signals are reached here to send a current through that, then it won't go all the way to my brain. Okay? And so this is the bipolar neuron that is found right here. So it's passing through the cribriform plate. And this is, as we'll learn tomorrow when we do our sensory pathways, this is identified as the first order. We have 3 sensory neurons typically. In smell we don't. But typically we have 3 neurons. So this is the first neuron. This would be the equivalent of what neuron in the spinal cord? The pseudo-unipolar. All right, so when I touch the table, and I activate my touch receptor, and that activates the pseudo-unipolar neuron, where does it go? It goes up my arm and to the dorsal root ganglion, and then into the posterior gray horn, right? So instead of doing that, the equivalent neuron for smell is going through the cribriform plate. That is really my olfactory neuron. Okay? Because my spinal nerve is coming into the spinal cord. This is the first neuron in the pathway, and it's synapsing with another neuron in the olfactory bulb. So when we have bipolar neurons, what we're calling the optic nerve is really just convenience for us. It's really part and extension, if you will, of the brainstem. covered with duramatter. It's the same neurons that we normally would see in the brainstem, not out in the periphery of the body. Okay? And so when we get an infection of the covering of the brainstem or the spinal cord, the meninges, that extends to the olfactory nerve. That extends to the optic nerve, because this is really essentially part of the brainstem or the spinal cord by structure. Okay? Now we ignore that and we just call it the olfactory nerve. But in reality, it's structurally something else. So here's our bipolar neuron. And then in the olfactory bulb, it's going to synapse with the second neuron in our pathway. So this is the olfactory bulb. And then it's going to create the olfactory tract, what we actually call the olfactory nerve. Okay, so I'm fine. I'm not going to mark you off if I say identify this cranial nerve and you say cranial nerve 1 olfactory. I just want you recognizing why functionally we get an infection or something else. It really is central nervous system. Okay. So, cranial nerve 1 is a sensory nerve. And of these 3 types of afferent neurons, it is going to be a special, visceral, afferent. For smell. Okay. So damage to that neuron is going to cause us a loss of smell, at least on one side of the nose. This, by the way, does not go back to the thalamus. It goes back to an area at the base of the parietal lobe where we process smell. Okay? And so it doesn't involve the thalamus for that relay center. So for those of you that just came in, we're going back to the cranial nerves we started on Tuesday and going in a little bit more information that's on the lecture notes. All right, so here's the olfactory bulb and tract, but if I say identify this cranial nerve, again, olfactory nerve is fine. All right. Now we're going to look at the optic nerve, and remember, vision uses the same bipolar type of system. So the optic nerve, what we call the optic nerve, is really the second nerve in our sequence and is really an extension of the brainstem rather than a true cranial nerve at the location where we mark it. So can I erase this part here? Okay. And again, these are the only 2 locations where we use bipolar neurons. Now again, most of what I'm going to draw up on the board is for free, because we actually do the eye in our next unit. I'm just trying to put this in sequence so you can understand why it's the differences between them and some of why they're there. So I'm just kind of, we've looked at this diagram before, I'm just going to blow up this part right here. Okay? So here is the back of an eyeball. And we have receptors. Just like we have receptors for smell, we have receptors for light. You've probably heard of them. They're called rods and cones. Okay? And their activity is changed by the energy of a photon. But they're just receptors. They work with the graded potentials, the rock that we're throwing in the pond. So we have those rods and cones along this, forming the deepest layer of the retina. And if they are activated by light, then a bipolar neuron is activated. And then from there, we have the second neuron. Remember in smell, the bipolar neuron went through the cribriform plate and synapsed with another neuron. If we were talking about peripheral nerves, that other neuron would have been the posterior gray horn. Okay? So now the second neuron, this would be the first neuron in our pathway. Now the second neuron in our pathway actually comes out of the cranial nerve, out of the eyeball, and passes back towards the thalamus. On the medial side, or nasal side, that cranial nerve is going to cross over. And that's this structure right here. Okay. Again, this is still for free. Okay? Just kind of looking at the pathway. We'll look at this in detail in our next unit. So, this is what we actually call the optic nerve. And you'll see it in the sheep brain. So leaving the eyeball is the optic nerve, but just like the olfactory nerve, it's really an extension of the brainstem. When we talked about the hypothalamus, remember I drew the hypothalamus for the embryology, a pouch growing out from the bottom and going out to the face? That's part of the diencephalon. These neurons came from the diencephalon, okay? So it really is still just part of the central nervous system, even though we're calling it a cranial nerve. It crosses, and then each of these is an optic tract. And these will go to the thalamus. So here they are going to synapse with the third neuron in our pathway. And that will go to the primary visual center. So is this a ganglion or a nucleus right here? Thalamus. It's inside the diencephalon, so it's inside the central nervous system. So it's a nucleus. The ganglion would be up here, okay, outside of the central nervous system in the retina. This is the thalamus, so this is the lateral geniculate nucleus of the thalamus. And then it ends up in our primary auditory cortex of the occipital lobe. Uh-huh.
Okay.
The thalami, the first neuron is the bipolar neuron.
Okay.
The second neuron is the one forming the optic nerve, which normally would be a spinal cord or brain stem. And the third neuron, as always, forms the thalamus.
It's not the number of synapses, because there are only 2 there, right?
Well, it's the first, second, third neurons, the number of neurons.
Right, not the synapses.
So primary visual cortex in the occipital lobe. So again, I've asked you to learn the lateral geniculate nucleus, so the reason I went through this pathway again is that's what forms it in the thalamus, are the cell bodies of the third neuron going to the occipital lobe. Yes?
Is the optic tract the third neuron?
No, the optic tract is still the second. Optic nerve and optic tract are still the second.
So what's the third?
The third is in the thalamus, going to the occipital lobe. And this is special visceral afferent for vision. And it's those are the 2 examples of bipolar neurons instead of pseudo-unipolar neurons. Yes, Tara?
These processes in synapse in the lateral geniculate ganglia of the thalamus?
It should be nucleus.
Okay, just to make sure.
Sorry, yeah. Should be nucleus. All right, so vision And then we move to the third. So let's go back here and look at the, I'm going to take a copy of